We are lucky we get 5 minutes extra, so we're going to inform you more about Dan Evan Roselle. Dan came to my lab when she was undergraduate. She's one of our students. She graduated from here with her BS, and before she completed her degree, she started doing undergraduate research. Dan is mother for 3, working part-time and going to go to school full-time. So full-time mother, full-time student, and part-time working. She managed to manage her time very intelligently. She has been working very hard in her thesis. What we're going to show you today, part of her thesis, not complete, is a part of her thesis and many other things she is in a process to complete. So what we're working with a plant, we call it Hibiscus plant, or we call it Roselle. A new plant being introduced to the United States back in '58, and very few people know about it. So in her approach, she's going to show you the commercial value of it, the health value of it, some analysis we done. So I hope you'll enjoy it.
Tell you a little bit about Roselle, because as Donnie mentioned, it's relatively unknown in this area. Roselle belongs to the Malvaceae family. This is the same family as okra, cotton, and cacao. It's an erect, mostly branched, annual, herbaceous subshrub. It grows mainly in warm and humid tropical and subtropical climates. It's native to India, but it's been exported to other areas such as Africa, the Middle East, just to name a few. A few of the common names, it has several, but a few of them in English-speaking regions is Roselle, sorrel, and red sorrel. In Arabic, it's called Coccidae. Roselle has a number of medicinal uses. It's been used in folk medicines for many years. Previous research has shown that a concentrated Roselle beverage lowers blood pressure in patients with hypertension and type 2 diabetes. It exhibits a mild laxative and diuretic effect, and a tea made from the leaves and flowers is used to enhance digestive and kidney functions. Sores and wounds can also be treated with a lotion made from the leaves. Every part of the plant is edible, and it has many popular food uses. Some of those are, as you can see up here, the calyxes can be chopped and added to a fruit salad. It can be stewed as a sauce or filling for fruit pies or tarts. Syrups, jams, jellies, relishes are made with the calyxes. And a drink that is very popular in many areas is, and can be served hot or cold, is also made with the calyxes. And I have brought some of that if anyone would like to try it. It's really pretty good. The young leaves and tender stems can be eaten raw in salads or cooked alone. As greens, they can be mixed with other vegetables and meat or fish. The seeds are very high in protein, and they've been ground to a meal for human consumption in Africa. The seeds have also been roasted and used as a coffee substitute. Hibiscus tea, which is the drink I mentioned earlier, is actually very popular, and it's a very old drink. It's said to have been a preferred drink of the pharaohs in ancient Egypt. This tea, hibiscus tea, contains 15 to 30% organic acids, including citric acid, malic acid, and tartaric acid. It also contains acidic polysaccharides and flavonoid glycosides, such as cyanidin and delphinidin, that give it its characteristic deep red color. And what we wanted to do in this research was, there's a lot of information out there about the benefits of Roselle. A lot of those haven't really been scientifically substantiated. So we wanted to examine some of those claims. And what we did was in order to see if maybe it should be promoted in this area, because it's pretty much unknown around here. We introduced Roselle to drought stress because we wanted to see how it would handle our summers here. You know, as many of you know, we had periods of little rain and high temperatures in the summer, so we wanted to see how well it could respond to drought and how well that it would recover from drought. We also evaluated the antioxidant production as well as the antimicrobial properties, and we had a variety of Roselle evaluated or analyzed for nutrient content in the stem, leaf, and calyces. And what we did was we grew the plants in the greenhouse. From seed, they were watered regularly until the pre-anthesis stage, and at that time they were divided into two groups. The control group received 100% soil saturation, and that was determined to be 2,000 milliliters. They were watered regularly. The treatment plants received 25% of this amount, and they were watered when they started to show signs of stress, such as leaf yellowing or leaf wilting. At the end of the third drought cycle, we wanted to know how the growth and flower production was affected by the drought. What we found when we did our analysis was that the drought had reduced the plants under drought conditions had undergone a 30% reduction in growth. However, the leaf water potential had only decreased by 18%, and this indicates that Roselle has the ability to combat stress. We also evaluated the photosynthetic pigment concentration, the chlorophyll A, chlorophyll B, and carotenoids of the two treatments, and we found that there was no significant difference between those two treatments. We also evaluated the total phenolic compound and flavonoid concentration. Flavonoids are a subgroup of the phenolic compounds. And what we found was there was no significant difference there between the control and the drought, which is something we thought we might actually see an increase in the drought plants, but the evidence showed us otherwise. And also at that time, we measured the photosynthesis rate, and as you can see, I'm not doing this right. Your far left, that's the rate that we took that day at the end of the third drought cycle. And you can see that there was a significant reduction in the rate of photosynthesis in the drought treatment plants. At that time, we divided the drought treatment plants into two groups. One of the groups was watered to 25% water saturation, the other group to 100% water saturation, and the control received 100% soil saturation. We took measurements of photosynthetic rate at 24 hours and 48 hours. And at the end of 48 hours, we found that the plants that received 100% soil saturation that had previously been under drought had recovered by 100% compared to the control. But those that had received 25% had only recovered to about 46%. In another experiment we did, we divided the drought plants into two groups. One group received 0% water saturation, the other group 25%, and the control received 100%. They were watered about once a week for 3 weeks, and then we looked into the antioxidant concentration of the calyces, and as you can see by the error bars that there was no significant difference in the antioxidant concentration of the calyces at that time. And in a separate experiment, we wanted to evaluate the phenolic extract of the calyces and see if they could be used as an antimicrobial agent against selected gram-positive and gram-negative bacteria. We used the disc diffusion method for that, and what we saw was that the extract had an inhibitory effect against Pseudomonas syringae, Staphylococcus aureus, Escherichia coli, and some inhibitory effect against Escherichia coli, but none against Staphylococcus aureus and Bacillus cereus. And in summary, what we found in doing this research, that the plant growth and flower production was significantly reduced by drought. The decline in photosynthetic rate induced by drought was recovered to 100% in plants that received 100% soil saturation after 48 hours. However, those that received 25% water soil saturation only recovered to about 46%. There was no significant difference in the photosynthetic pigments or the phenolic compounds, And we found that Roselle had an inhibitory effect against Pseudomonas and S. aureus, and some against E. coli, but none against Micrococcus roseus and Bacillus cereus. In a separate experiment carried out at the Field Experimental Station at Auburn University, we looked at two varieties of sorrel, the red drops and the Thai red. We wanted to see if maybe one should be promoted above the other. We looked at gas exchange measurements between the two varieties and found there was no significant difference between those two varieties. We also looked at selected pigments and anthocyanins, and again there was no significant difference between those two varieties. We ended up having the Thai red variety analyzed. We had the stem, leaf, and calyces analyzed for nutrient content. The results of this analysis showed that crude protein was highest in the leaf. Neutral detergent fiber was highest in the stem, and also the stem was highest in acid detergent fiber. And the calyces was had the highest percentage of total digestible nutrients. And the significance of that is that currently Americans aren't really ingesting the recommended amount of fiber, and they're spending a lot of money on these fiber substitutes that contain psyllium, and they really have no nutrition or very little nutritional value. And so Roselle would be a good source of, from our research, we thought Roselle would be a good source of fiber. We divided the micronutrients, the minerals, into microminerals and macrominerals, and the highest percent of nitrogen was found in the leaf. The highest percentage of potassium was found in the stem and calyces. Phosphorus, there's really no significant difference in those between stem, leaf, and calyces, or in the magnesium. The sodium was highest in the stem, there was no significant difference there, and calcium was highest in the leaf. I'm not sure if I mentioned calcium. We also found that iron was highest in the leaf, magnesium was highest in the leaf, zinc, there was no significant difference there, and copper was also highest in the leaf. And the conclusions we drew from this research was that the calyces, followed by the leaves, can be an excellent source for animal consumption because of their, due to its ADF and NDF values. For human consumption, all parts of the plant can be considered an excellent source of fiber. And in general, this plant appears to be relatively high in micro and macronutrients. And I wanted to thank Ms. Tia Gonzalez, Auburn University, Ms. Hannah Leggett, Mr. Richard Watkins, Dr. Dennis Shannon, and Jacksonville State University for all their help with my research.